the day today about someone needs an override for class. I'm like, it sounds like you probably do. Good luck. Yeah. 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 I have the, what is it, Beaver Hub, the advising one? Yeah. I have that, but I have no power to do anything, but I can look up people's names. Like, that's pretty good, I guess. Yeah. Yeah, they have all like so all the grad students are listed in there for me because I'm technically their advisor. Yeah. And so I can help them like figure out issues, but like I can't give you an override. Who do you think I am? Like I don't have power. Yeah. Oh, that's funny. Okay, sounds good. Yeah.
I think we got a lot of crack.
Yeah. Yeah, that's good.
All right, we can go ahead and get started. So thanks for coming and thanks to our speaker. So Nate Mortimer is actually 2 floors down from here.
Yep.
So he's an associate professor in biophysics and biochemistry. He's been at OSU since 2022. Prior to that, he had faculty positions at the University of Warwick and Illinois State University. And then prior to that, he did his PhD and postdoc for training at Emory. So Nate is sort of broadly interested in cell signaling and immune function. And he has a really cool system of Drosophila and these different parasitized wasps he uses to answer questions about that he's going to be talking to us today.
Yeah.
So thanks, Nate.
Yeah, awesome. Yeah, thanks, Mark. Thanks everyone for hosting me today. It's been a really, I was gonna say trip, but across the hall to Cordley, I've really enjoyed myself, and it's crazy. I've been here for a few years now and not met very many people in your department. So thank you all for showing up today and for coming out to meet me today. It was a really fun time. So yeah, so like Mark said, in the lab, we're interested in looking at cell signaling. We're interested in looking at host-parasite interactions. And so I have my fun title here of Invasion of the Body Snatchers, and you'll see exactly how on point it is as we go along. If you have questions along the way, please feel free to interrupt me. I'm not a formal speaker. I don't, practice and memorize. So we're just going to wing it and see how it goes today. Okay. So like I said, we're interested in thinking about host pathogen interactions in the lab, right? And at its most basic level, we can think about hosts mounting immune responses to eliminate pathogens, and then pathogens being selected to be able to successfully target their hosts. But it's a little bit more complicated when we start thinking about what that actually means, right? So we think about immune responses, right? They get induced by infection, so we're not chronically in an immune state. Well, most of us. The immune cells have to recognize and respond to the pathogen, and then there needs to be this metabolic reallocation to fuel that immune response, right? So the host is doing a lot just to be able to mount this immune response in the first place. And pathogens, of course, don't want any of that to happen. And so they've evolved these virulence factors that allow them to manipulate the host signaling. And these virulence factors target things like host metabolism, the host immune response, as well as their development. And so I'm really interested in studying sort of how these systems work and co-evolve. And so when I was thinking about getting into this research, I kind of wanted to design in my head a system to use experimentally, right? And so my ideal model host is an organism where we can look at genetic and biochemical mechanisms and then test those mechanisms at the level of the organism. And so it's kind of obvious who I chose, but Drosophila is great for this kind of research. And then thinking about the pathogen is also kind of a difficult thing. And so I wanted a pathogen that naturally co-evolved with its host, but then also one that we could maintain in the lab and was easy to do experiments with. And so then I stumbled on these species of parasitoid wasps. And we'll talk about the wasps in just a second. I'll give you a bit better introduction to them. This is just to show how cool they look for the people on Zoom. I always think it looks like they have motorcycle helmets on, and they look like bad guys from like 80s action movies. So I'm a big fan of the wasp. Okay, so we'll start with the host. So Drosophila melanogaster, I'm sure a lot of you have seen something similar to this before. They are great to use as hosts. We have a really wide range of molecular tools, genetics tools that we can use to do experiments. They have a lot of highly conserved signaling mechanisms. So we have lots of human disease models that we study in the flies. And they have an innate immune response that's mediated by really conserved signaling pathways as well. So not just basic biology, but specifically this immune response that I want to look at. They have a well-conserved version across evolution with us and with other insects and organisms of interest. And importantly, flies get naturally infected by a really wide range of pathogens, right? So just like us, they can get viruses, they can be infected by microbes, and they get infected by parasites. And each of these pathogen classes is recognized differently, and they mount a specialized immune response against. And so I'm sort of started life as a cell biologist. I'm really interested in how cells function. And so I wanted to look at an immune response that was mediated by cell mechanisms. And so that is how I got into looking at parasites, because they are targeted by a cellular immune response. All right, so if we get a little look at the parasitoid wasps themselves. So here's another beautiful picture from Alex Wild. These wasps are obligate parasitoids of Drosophila larvae, the ones that we study infect the larvae. During infection, they lay their egg directly in the hemocoel of the host, and they also transfer in their virulence factors in the form of what we call venom. So they transfer this venom into the host, and it becomes this really interesting system where wasp venom proteins are essentially combating host immune molecules to figure out who's going to win this infection. So it's a really nice little closed system. These wasps can be very easily propagated in the laboratory. So if you can maintain flies, you can maintain wasps. And they're really tractable for a lot of types of experiments that we like to do. So we do infection assays, we do all sorts of omics, and we do all sorts of biochemistry with the proteins that we isolate from them. But everyone wants to see a wasp in action, and so let's take a look. So here we have a female parasitoid wasp, and here's a little fly larva trying to escape. If we look at this structure right here, is the ovipositor. And so this is what she's going to use to infect this fly larva. And so if we let the video play, you can see it's a very quick process. The ovipositor pierces the cuticle of the fly. She deposits the egg, inserts the venom, and essentially pulls out and is done, cleans off the ovipositor, wanders off to find another host. So it's a really cool little thing to do and watch them for half an hour every time I set up an infection because it's just really fun to watch. But that's sort of how the system works. And so this is just obviously the beginning of it for this poor fly that's been infected. It now has to deal with all the terror that we're about to talk about. So like I said, the outcome of these infections is determined by the interaction between host immune molecules and parasite virulence. And in the lab, we have lots of different species of wasps. Among these are what we consider avirulent species. So they're avirulent with response to Drosophila melanogaster, which is our main host, meaning they can't successfully infect melanogaster. And throughout this talk, when I talk about an avirulent wasp, it's a species called Leptoplanet clavipes. Not that that's important, but in case anyone is interested. And these wasps actually normally live on mushroom-eating flies. So they are not very good at infecting Drosophila melanogaster, which are not mushroom-eating flies. And then we also have all of these species of virulent wasps, so wasps that can successfully infect Drosophila melanogaster. We're going to talk mainly about one species today, Ganaspis hookeri, which I always just call GH. So if I say GH, that's who I'm talking about. And these are great because they have active venoms that we can study how they manipulate host signaling. All right, so here's the artist's rendition of what happens. So we'll just sort of think a little bit about, there's an infection, right? So wasp comes, has transferred the egg and the venom into the host. So what happens next? So something that our labs found that I think is really interesting is immediately after infection with a wasp or with any other pathogen, there's this burst of what I consider sort of the immediate immune signaling molecules. So transcription of things like antimicrobial peptides, pattern recognition receptors as kind of a generalized immune response. And then this gets followed by a response that's very specific to the infecting pathogen. So in this case, it's an anti-parasite response. So we'll take a look again, the artist's impression of how this works. And in this case, the artist is one of my former students, Ashley Waring Sparks, who spent a lot of time on this for her dissertation. So I promised her I would use it every chance I got. And so what we're looking at here is the fly life cycle. And we were looking specifically at larvae and sort of the late phase in this middle here, sort of mid-sized larvae. And then a wasp comes in to infect them. It deposits the egg and the venom into that host. And so if we kind of zoom in, we can see all of the events that happen after infection, right? So if we look at the egg in isolation, it's just laid into the host hemocoel. Flies have an open circulatory system, and so all of their immune cells are kind of washing around this egg. And so what we're showing here are a class of immune cells called plasmatocytes. These are their most abundant immune cell. They act essentially like macrophages. And after infection, they get activated. And we'll talk a lot about what that means later because it's something I think is really interesting. And then after activation, they then express the pattern recognition receptors that allow them to find the wasp egg and interact with it. So these plasmatocytes start going to the wasp egg, and eventually they form a layer completely over the egg. Nothing bad happens to the wasp when this happens, but this is sort of the start of the immune response. And then at the same time, the fly starts differentiating a second class of immune cell, and these are called lamellocytes. To scale, these are probably 4 or 5 times bigger than a plasmatocyte. They are these great, huge, thin cells with long actin projections, and the only thing they're good at is sticking to stuff. And so you can imagine how good this is when you're trying to mount an immune response against a specific target. These lamellocytes are targeted to the plasmatocyte layer, and they form an outer layer around this wasp egg. This layer goes through what we call consolidation. So there is a protein glycosylation driven change in the cell-cell adhesions, so it becomes completely impermeable. After it becomes impermeable, the cells melanize. And so they are producing reactive oxygen species that they're dumping into this wasp egg. They're forming a layer that cuts it off from circulation, so there's no oxygen or nutrients getting in. And so essentially they have now choked out this wasp. And what it looks like is this. So in the fly itself, we can actually see this as this little melanized body that's left behind. Of course, mounting an immune response is costly, right? So if you're a host thinking about mounting an immune response, you're going to consume a lot of energy. You need to increase your transcription and translation of new proteins. You need to produce cells, so you need more lipids and all of the business that goes along with it. And what we find is that after infection, these larvae have this shift in metabolism. And we can read this out as changes in metabolite abundance. And so, yes, I snuck a little bit of data into the introduction, and I apologize, but it's cool and it's new, so I wanted to talk about it. So we did some global metabolomics and compared flies that were infected to naive. And what I'm showing here is a volcano plot. So above this line are all the cells, all the metabolites that are significantly changed. To the left are ones that have a reduced abundance after infection, and to the right have a greater abundance after infection. And so the first thing and this is just a handful of them, but these are cool, so I wanted to highlight them. So the first thing we notice is that infection alters glucose metabolism. And what we actually see is that glucose is being diverted away from glycolysis in the TCA cycle, so all the things in red. And instead it goes through the pentose phosphate pathway, which is the things here in green. We also see that infection changes lipid synthesis. So we see the appearance of a bunch of lysophospholipid, which are not present in a naive fly. And what's cool is that these changes are actually broadly conserved. So when we get infected in our macrophages, these same 2 things happen. So it's a really cool system, I think, to start looking at how this metabolic reallocation happens, and we will in a couple of minutes. But what I want to do first is sort of complete the story and think about this from the wasp perspective, right? So I did my grad school in a fly lab. I thought when I was done, I would never work with flies again. And then I got the chance to work with something that killed and ate flies. So I thought that was kind of a cool move to go down. So I did that. So I like to think about things from the wasp perspective. And so the wasps, of course, don't want any of the things that I just told you to happen, right? And so what they do is they have these virulence factors that they've evolved in their venoms that allow them to overcome the host responses. And so, like I said before, these venom proteins have a wide range of functions. Specifically, the ones we're going to think about are preventing activation of the host immune response and also manipulating that host metabolism. And for people who are graphic, that pathway I showed you before, we found venoms that block every single step in it across different wasp species. So at the activation, at cell migration, at cell adhesion, at melanization, can be blocked by venom proteins. And if it's blocked at any of those stages, the response fails. And then we get, this is our little wasp larva here that hatches out from the wasp egg and will eventually grow up to be an adult fly, or adult wasp, sorry. Now, these venom proteins are really, really important. So we want to talk a little bit about where they come from. And I also want to show a cool image. Hopefully you can see that okay. If not, we can change the lights. This is a venom apparatus that has been dissected from a wasp and stained with phalloidin. So this shows all the filamentous actin in the tissue. We can sort of break it down into a few specific tissues. So if we look here is the venom gland. These things all have really great names. So the venom gland, and these are the cells that actually do the transcription and translation of all of the venom proteins. Those proteins get secreted, and we'll talk a little bit about how they get transported later, but they get carried down this collecting duct here and dumped into the venom reservoir. And if you look at it, you can see all of these striations, and that tells us that this reservoir is actually surrounded by muscle. And this reservoir lies right next to the ovarian cavity. And so this tissue is connected, if we think way up here off screen, is where the ovaries are. And so the eggs, one at a time, get transported down into this cavity. We're a little bit out of focal plane, but you can see this is also surrounded by muscle. And you can see the outline of a wasp egg sat here ready to go. Both the ovarian cavity and venom reservoir dump out into the ovipositor here. And then this is their ovipositor, which for scale probably ends a floor and a half above where we are currently. But that's the ovipositor, that's what they're sticking into the host. This ovipositor has a bunch of sensory neurons with their axons that run all the way back down the ovipositor. And when the ovipositor encounters the internal environment of a host, that triggers these neurons to fire, sends an action potential the length of the ovipositor, and the venom reservoir and the ovarian cavity contract. And that's how you actually get the egg and the venom transferred over to the host. And I've always wanted to do some neuroscience with the ovipositors, but not yet. We'll see how that goes in the long run. Okay. And the thing that's nice about all the venom proteins being held in this one place is it's very easy for us to purify the venoms and do experiments with them. And so what we can do is dissect out the venom apparatus, the glands and the reservoirs, and purify just the venom away from the rest of the cells. And then we can identify what those proteins are. And we've done this so far for 3 species. We have a couple more that as soon as we get money, we want to do, but we have a few done. The venoms turned out to be a lot more complex than I thought going in. So depending on the species, we're thinking somewhere around 150 venom proteins. And then we've also assembled transcriptomes and now genomes for these species as well. And these are the collaborators who have all the expertise to do the omics parts that I was just learning at the time as a young postdoc. So we can sort of move on and think about some of the big research questions, right? So I'm kind of interested in thinking about how immune systems work. And so one of the big questions is, how are immune cells primed for an immune response? Also questions like, what mediates this metabolic reallocation after infection? And is that involved in life history trade-offs? And then, of course, from the wasp perspective, how do their venom proteins or their virulence factors inhibit these host immune mechanisms? All right, so we'll start out and we'll think a little bit about immune cell activation or immune priming. So I was very surprised to learn when I first started learning about immunology that immune cells in a naive organism are not competent to do immune responses, right? Like they exist to clear away dying cells and all sorts of things, but they don't actually have the goods to be part of a response to infection. So they need some kind of priming. And that's something I've gotten really interested in. And this priming, which we call immune cell activation, results in production of immune recognition receptors, of cytokines to trigger signaling. And conserved across species is this mechanism in which increased intracellular calcium signaling leads to cell activation. And so we're going to start with the hypothesis that intracellular calcium signaling mediates immune cell activation in our parasitoid infected flies. If that's true, we would predict that immune cells would have increased cytoplasmic calcium following infection, so we can infect flies and use a readout of calcium concentration to test this prediction. We'd also predict that if we block this calcium signaling, we'll decrease their immune success. So we'll take a look at a couple of experiments to test these predictions. So first we need to talk about how do you look at calcium signaling in a cell. So how we do it is express a fluorescent calcium sensor in the immune cells. So this is GCAMP3 is what it's called. It's a version of GFP that is, when it's translated, is inside out. It needs calcium to bind to it to put it all back together. So whenever calcium binds, you get fluorescence. When it's unbound, there's no fluorescence. We can express this in immune cells to look at calcium levels, and the amount of fluorescence is relative, right? So if we have no calcium versus some calcium, we'll see this no GFP to some GFP. If levels of calcium go up, then the levels of fluorescence go up. So we can use fluorescence intensity as a fairly easy readout of cytoplasmic calcium. And so we do this. I think it's going to be a little bit tough to see up here, but I'll try to point you in the right direction. So here we have a field of immune cells from a naive fly, and they have very, very faint GFP. We can quantify it. I'll show you a bar graph in a minute, but cells are cooler than bar graphs, so we'll start with the cells. When these flies get infected with an avirulent wasp, so a wasp they'll mount a successful immune response against, we see a big increase in the amount of fluorescence, so in the amount of calcium that's present. So prediction 1 is good. We also want to test if mutations that decrease intracellular calcium levels are going to lower their immune success. So here we have a control fly. Sorry, I want to point, but I know I'm supposed to use this thing. So we have a control fly, and it's successful about 90% of the time it gets infected. If we manipulate this host to have even just slightly less calcium, that success significantly goes down. Unfortunately, because calcium is so important, we can't abolish it. But even just slight decreases in calcium make the flies have less immune success. So it appears as though both of our predictions are true. So that leads us to a question, and there's been kind of a little bit of circular logic in what we've been talking about, right? So immune cells need to be activated in order to be part of an immune response, but they get activated after infection, so how do they sense that? And so the hypothesis is that rather than immune-specific molecules, they're actually recognizing what are called DAMPs, damage-associated molecular patterns. And so DAMPs are molecules that come from the host tissue following infection, so damage or stress will lead these to be released. And a lot of times extracellular purines, so adenosine, guanosine, are really common DAMPs that we see in immune responses. And so if purines are the DAMPs that are involved in this immune cell activation, we'd predict that if we treated immune cells with purines, we would see an increase in calcium level. We'd also predict that if we block the purine receptor, we would block that calcium-mediated response. And then finally, we would predict that the purine levels will increase in flies after they get infected in order to serve as a signal. So let's take a look through these things. So the two DAMPs that we're looking at are extracellular adenosine, which you'll see as E8O, and guanosine, E-GUO. So adenosine and guanosine. And so we first wanted to ask if treatment with either adenosine or guanosine increased calcium levels in the immune cells. And the answer is yes, right? So looking at this plot, the dotted line here at 1 is the control that we've normalized calcium levels to. And we see a fairly consistent increase with either treatment. You can ignore the numbers here and just look at the data distributions. The numbers are arbitrary, but you see quite a solid increase in the amount of calcium if we expose the immune cells to these purines. The next question is whether we can block this by blocking the purine receptor. So we have an antagonist to that receptor that we can pre-treat the cells with, and then add the purine and see what happens to calcium levels. And when we do that, we actually either return to baseline or even get a little bit below baseline with the amount of calcium. So it certainly seems as though purines are signaling through a purine receptor to these immune cells to change their calcium activation state. I have an undergrad student who's working on this, and right now she is looking at mutants that are unable to make these extracellular forms of adenosine and guanosine to see if that decreases their immune success, which we would predict based on this. The final prediction is whether we see an increase in levels of purines after infection. And so we'll first look at adenosine here. So we're looking at naive individuals at time points early after infection where we see this immune activation, and then we're looking late when immune activation is done. When we look at adenosine, we actually don't see a change, which was really disturbing, but that's the way it goes. But if we look at guanosine, we see something different, right? So here's guanosine in the naive state. And when we look after infection, we see a significant increase in guanosine abundance, specifically at the early time point when immune cell activation is happening. So we can kind of come to some conclusions that it seems as though this extracellular guanosine is acting as a damp to induce intracellular calcium signaling, which leads to immune cell activation. But of course, the interesting part is, can the WASP do anything about this, right? Is there a WASP that's able to block the activation of immune cells? And so obviously we think there are because we started doing the project. So we hypothesized that there are virulence factors that can block this activation step. And we would predict that this would be correlated with a parasitoid infection that prevents this increase in intracellular calcium levels. We can also predict that if we can genetically increase the amount of calcium in immune cells, that should confer resistance to the WASP. And so we're using our favorite WASP here, GH, Ganaspis hookeri. And when we look at its calcium levels in immune cells using the same trick we did earlier, so again, our control with minimal calcium. Now if they get infected by GH, we actually see even the minimal fluorescence is gone. So GH venom is very effectively eliminating calcium signaling from these cells. And I promised a bar graph, and here it is. So if we look at our early time point when immune cell activation is happening, we see with an avirulent infection a pretty strong increase in the amount of calcium levels within the immune cells. GH infected, that goes way down. At that late time point when immune cell activation is done, everything kind of comes back to baseline. So we see this ability of GH to decrease calcium levels, but is it sensitive to calcium? So we used a couple of mutations that would increase the amount of calcium in a cell. And so there are technically bars here. So the two background control lines are not able to, they have zero resistance against this WASP species. So I have looked at tens of thousands and seen one or two successful immune responses. So they are very, very not incapable of responding to this WASP. But if we make genetic manipulations where we add back just a little bit of calcium again, we see even up to 50+% success rates. So these WASPs are very sensitive to the amount of calcium signaling in the host cell. So it seems as though GH is able to block immune cell activation at the level of blocking this increase in calcium. But I'm always driven by the question of how. I always want to know how something happens. And so I wanted to know how this was happening, right? How is the venom able to block calcium signaling? And so I said we did mass spec to identify all the proteins. There are 160 of them. But of course, you always put them in order of abundance and then start looking down the path or down the list to see what might be cool. We didn't have to go very far down the list before we found something cool. And that's this protein called SERCA, which stands for sarcoendoplasmic reticulum calcium ATPase. And what all of that means is it's a protein that pumps calcium against its concentration gradient. And essentially what it does is it lowers the amount of calcium in the cytoplasm and gets it out of cytoplasm and into the store. So it basically blocks intracellular calcium signaling. So we thought it was very cool that we found this protein in venom. We did some experiments that I'm not going to get into, but the takeaway is that if we treat venom with a protein or a compound that blocks SERCA activity, we completely block the ability of venom to inhibit calcium signaling. So this SERCA pump is somehow getting into immune cells, decreasing the amount of calcium that's in there, and that is leading the fly to essentially be blind to the fact that they've been infected. But again, there's another level of how, and now the how comes from looking at the SERCA protein itself. So when we think about insect venoms, generally we think about small peptides. That is not the case here. SERCA is a huge protein. It has 11 transmembrane domains, so it's very hydrophobic. So here is sort of the membrane. So here's all the hydrophobic area, and then this is hydrophilic. So there's very little chance that this protein is just being secreted into aqueous venom and happily making its way through the fly hemolymph to find an immune cell, right? So we think there has to be some kind of packaging. Other parasitoid species use extracellular vesicles. So venom extracellular vesicle, or VEV, is an abbreviation you'll see a lot while we're talking about this. And the idea is they can use these vesicles to transport their venom proteins. And so we hypothesize that a similar mechanism is how SERCA is getting into the host immune cells. And here we have a very simple prediction that we're going to find something that looks like a vesicle if we look at venom. And so that statement, if we look at venom, made our lab learn a whole bunch of new techniques. So we learned how to do electron microscopy. We learned a bunch of biophysical techniques to characterize nanoparticles. And so I'll show you some brand new data that I'm really proud of that we were able to collect. So first we have some electron microscopy looking at whole venom from GH. So first we'll look over here at a scanning electron micrograph, and you can see there's a lot of particles in this venom. They range from really, really small. So you see some of these really small particles here. These are less than 50 nanometers, which is too small to actually be a lipid enclosed particle. So we think these are likely large protein complexes and most likely lipoproteins. And then we see all these things sort of from 50 nanometers up to 150 nanometers maybe that look like they could be vesicles. We did some negative stain transmission electron microscopy over here, and yep, they sure look like vesicles to me, right? So we see a bunch of these vesicles. What's cool is that in all the other species that people have studied, these vesicles are very, very uniform, right? So they're only make one size, they all have the same density. They haven't really studied anything else about them, but they all look the same. And ours certainly don't. And we can go in a little bit closer and look at some of these vesicles and see that we have big size differences, right? So the one on the right here is about 2.5 times the size of this one. And that's not the biggest difference because it has this weird, almost kidney bean morphology compared to the beautiful round classical vesicle we see here. And we see these in great abundance. So this is not the one weirdo that we found. We see a lot of this class of vesicle. So we started thinking maybe we have a polydispersed population of vesicles. And so we used a technique that would let us look at the diameter of the vesicles and their concentration. And so if we plot a histogram, you can see it looks but definitely like there's more than one population there. So we did some mixture modeling to have a look at what's going on in this population, and it predicts 3 populations with distinct sizes of vesicles, and you can see the 3 sizes labeled here, which kind of spans what we see from our EM. We did a whole bunch of other biophysics that I won't bore you with, but we looked at diameter, we looked at the surface charge on the vesicle, we looked at the density of the vesicle, and then of course you have morphology from the microscopy. And it looks like there are anything from 3 to 5 different categories of vesicles that we find in this venom. And they vary across all of those characteristics. So we're really interested in starting to uncover all of these vesicles and how they function individually. And then just for comparison's sake, the 2 other species that people have found vesicles in, one of them is huge and looks like a virus. So it has spike proteins in the whole deal, but they haven't characterized anything else. The other are kind of in the same size range, but they have, again, a weird morphology, so are quite distinct from what we're seeing. And a lot of ours look a lot more like classical extracellular vesicle derivation. So that's great, but are these vesicles actually required to do anything, right? We can find them, but are they functional? Right, so our hypothesis is that these vesicles are transporting venom proteins like SERCA into host cells. If that's true, we would predict that the vesicles will actually be able to enter into the host cell. We would also predict that if we disrupt the integrity of the vesicle, that will get rid of the activity of the venom. So a couple of straightforward experiments, it sounds like. So first we'll talk about how we can look at vesicle entry into host cells. And so what we use is a lipid dye that we can label our venom with, and then we can quench that dye so there's no free dye to bind. And so if we just use a buffer control rather than venom, what you're looking at on the left here is the bright field image of a whole bunch of immune cells. On the right is the fluorescence that should come from this lipid dye. And you can see it's a fairly clean background, right? There's a couple of little spots of fluorescence, but not a whole lot going on, right? So it seems as though we're not getting a lot of background signal when we use this lipid dye. But when we label venom and put it on the immune cells, we see something completely different, right? So not all of the immune cell subtypes, but most of the immune cells end up with label being taken up into them, suggesting that these vesicles are gaining entry in some kind of a way. We can look at a higher magnification and do some pseudocoloring to look at fluorescence intensity. And when we do this, we actually see that these immune cells are taking up vesicles in what look like punctate structures. And so we have a hypothesis that they're actually being endocytosed by the host cells. We are in the process now of knocking out all of the different endocytic mechanisms to see if any of those prevent uptake. Preliminarily, it looks like we might be on the right track, but we'll have to see. This is my student Nick Bretz, who came with me from ISU here. He's graduating soon, sadly, but we'll see what he finds in the next couple of months. The other prediction we made was that disrupting the integrity of these vesicles should block the activity. And so we can use our calcium assay to get a sense of this. And so now here we're looking at control cells, so cells that were incubated just with buffer, compared to cells incubated with venom. And we see this decrease in calcium levels within the cells treated with venom. And so what we wanted to do was 2 treatments to the venom. The first we did heat denaturing, so we boiled the venom so we would denature all of the proteins, so there should be no activity. And then we took the venom and sonicated it. And so the idea here is this should disrupt any structure, any lipid-bound structures, but won't disrupt the proteins themselves. So the proteins are active, are present, but they're not found in a vesicle anymore. And if we look at, that's what I just said, if we look at their ability to decrease calcium levels, we see that this largely blocks, both treatments largely block the ability of venom to decrease calcium levels in immune cells. And actually sonicated now becomes indistinguishable from control, suggesting that these vesicles are required for SERCA to get in and do its job in the immune cells. And so if we kind of wrap up this section, We talked about how host immune cell activation is mediated by DAMPs and calcium signaling, and that the parasitoid Ganaspis hookeri uses circuit-containing extracellular vesicles to inhibit host calcium signaling and block host immunity. But I promised you one more story, and that is trying to figure out what mediates the metabolic reallocation and life history trade-offs after infection. Right? So this is a question that I think is really exciting, really interesting to start thinking about. So I was very happy when I finally got a chance to work on this. And so to kind of give you the background that you need, right, we talked previously about immune cell activation and how it leads to production of cytokines. So one of the cytokines that we found is called PVF. So this is ancestral to platelet-derived growth factor, vascular endothelial growth factor signaling in mammals. This pathway has been previously linked to metabolism, both in flies and in mammals as well. And so we hypothesized that the PVF cytokine signaling that originates from immune cell activation is mediating this metabolic reallocation. And so we can make some predictions. So first we would predict that if we disrupt PVF signaling, that would impair the host immune response. And then we'd predict that if we could ectopically turn on this pathway, it would cause the metabolic shift independent of infection. And so we take a look at these things. Right, so first we're looking at knocking out PVF signaling pathway and its effect on host immunity. So you can see both graphs here are obviously showing that manipulating the pathway leads to decreased immune success. On the left, we are using RNAi to knock out the ligand, the cytokine itself. On the right, we're expressing a dominant negative form of the cytokine receptor. So either way of knocking out this pathway leads to a big reduction in immune success. And so we got started on this whole thing. My student, Ashley, is the one who started this. And for the grad students, I'll sort of tell you the behind the scenes story of how we got here and why it's important to really learn your system, right? So we started out with this hypothesis about PVF signaling, that it was going to be what dictated migration of immune cells, right? So it's been known for a long time that this class of signaling factor leads to cell migration. And so we set up this wonderful experiment to test cell migration. Ashley did it and saw no phenotype. She did it again, she did it again, she did it again. And of course, it has nothing to do with cell migration, but in the process of doing this experiment and really sitting and looking at her mutants, she realized something very simple, but very fundamental. And that was when she was dissecting the mutant flies, her buffer did not get cloudy. And she came and told me, like, she was like, what does this mean? Like, I've done this 1,000 times and the buffer does not get cloudy. And what we realized is the cloudiness of the buffer comes from dissecting the larva and seeing fat float away that had been released as part of this metabolic reallocation. And so her buffer was clean because there was no reallocation of metabolism, so the lipid synthesis was not changed. And so we completely changed her thesis project, and it's all because she sat there long enough to realize that something looked weird when she was doing the experiment. And it had nothing to do with science, it was her buffer, but that led us into figuring out this whole pathway and how it functions. So I always think that's kind of a cool story and a little bit inspiring that you can be wrong and still figure out something worthwhile. Okay, so our other prediction was that ectopic PVF signaling would phenocopy this metabolic reallocation. And so what we're looking at here are naive flies compared now to flies that are expressing a constitutively active form of the cytokine receptor, right? And so what that means is these immune cells are always getting cytokine signaling active, even when there's no cytokines around, right? So in the absence of infection, the cells think they are because they're having the cytokine signal. And when we look at the metabolites that get changed in the two genotypes, we see that there is a real significant overlap of metabolites between our constitutive PVF flies and our infected, so our immune-induced flies. And so it seems as though this cytokine signaling pathway is actually contributing to this metabolic reallocation that we see after infection. And then, of course, this leads us to start thinking about, oh, that's very jumbly, leads us to start thinking about life history trade-offs, right? So this is something we've all kind of learned. Immunity is costly, things get reallocated, and that leads to decreases in fecundity, survival, development rate, whatever it is that you want to think about. And so since we're looking at immune responses in immature flies, we first wanted to look at development. And so this is just a little window into fly development, going through the late larval stages, going through pupal formation, metamorphosis, becoming an adult fly. And so what we did is we looked at our 3 genotypes. So our control, if we block PVF signaling or if we activate PVF signaling. And we saw how long it took them to get to each stage and how many of them got to each stage. And we found there are delays in their development. But even more impressive is that the flies that are expressing the activated cytokine receptor, so the ones in magenta here, have a lot less of the flies are making it to each developmental stage, right? So they're mostly making pupae, so they're getting there. We have a significant amount that are dying as they're trying to go through metamorphosis, and then over 40% of them can never actually eclose, so they can't get out of the pupal case as adults. And so we have a pretty severe, what appears to be a trade-off between ability to complete development and to mount an immune response. This is obviously a case where it's a little overblown, where we're expressing this activated cytokine receptor all the time in these immune cells. So we want to start doing some temporal shifts to look at different windows of activation to see if we can look at life history trade-offs without the actual infection part. If we can just genetically manipulate the signaling pathway, to understand what happens. All right, so we can wrap this up. So I told you 2 stories, well, I guess 3 stories today. The first about immune cell activation. So after infection, these DAMPs, damaged particles, are released into the hemolymph. Those are sensed by the immune cells, which leads these immune cells to become activated. The wasp Ganaspis hookeri is actually counteracting that by using a calcium pump found in its venom to pump that calcium back out of the host cell and block immune activation. And then we talked about the PVF cytokine pathway, which mediates metabolic reallocation and life history trade-offs after infection. And so the most important slide of all are the people who did all of the things. So we'll point out a couple of people. This is not everyone, this is sort of a little group. So we'll point out Nick and Chris are the 2 students who figured out all of the vesicle story. Ashley and now Abe are the 2 students who are working on metabolism and this PVF signaling pathway. And then Hayden here is an undergrad who's figured out how adenosine and guanosine are contributing to the story. And a lot of these other people contributed to a lot of things that I would need a whole day to talk to you about. So we'll stop it there. And I'm happy to take any questions that anyone has. Yeah. Yeah, thanks. Yeah, good.
Is it just an amount of venom each time, or does it vary as part of that influence the?
Yeah, that's a really good question. So I'm notorious for not giving a straight answer. So I'm not going to give you a straight answer. I'm going to give you the complexity of the answer, right? So when we think about the idealized infection, yes, they're very consistent in the amount of proteins, the ratio between all the different proteins they're putting into the host. They are, of course, animals. They get old, they get tired, they get raggedy. It probably doesn't stay the same across their entire lifespan. But that's not something we've ever really been able to characterize other than the fact that they do use up venom before they use up eggs. So they probably are probably shooting blanks by the time they get really old is way I would think of it. We also did an experiment that I think is really cool. So I didn't talk about host range very much. Some of these wasps have very restricted host ranges, so we call those specialists. Some can infect really broadly, and we call those generalists. And we took one of our generalist wasps, and we being Dakota, who is not on the slide, but Dakota, another student that I had, was really interested in this idea of sort of plasticity, evolution of the venom. And so he took generalists and raised them on different host species for several generations, and then looked at their venom composition. And it actually changes based on the host that they're living on. So there's some plasticity to it. And when they're on the same host generation after generation, in some cases, there were actually what appeared to be different venom proteins, but a lot of the time it was just a different combination of the venoms. So something that was not expressed very highly became expressed much more highly, that kind of thing. So there's a lot going on there, and it's a really interesting area to get into. Yeah, thanks for the question. Yeah. Yeah, so okay, that's a great question. So the wasp egg hatches while the fly is still a larva. So you have a larva inside a larva situation for a day or two. The wasp needs the fly to pupate because the wasp cannot make their own pupal case. So they're almost doubly parasitizing, right? They're taking their nutrients and everything else, and then they're making use of their house once they eat them. So it's a little bit, invasive, I guess. But yeah, so they wait for the fly to pupate, and it's while the fly is going through metamorphosis, that's when they actually start eating the fly's tissues. So they consume the fly, then they complete their own metamorphosis inside the fly's pupal case, and then they chew a hole and fly away. So it's really, yeah, it's pretty cool. Yeah, yeah. So in some populations, 90 plus or 80 plus percent of flies can be infected. And so they do have some success, or they wouldn't exist anymore. And when we look at it in the lab, we're generally looking at or wasp lines that are either really virulent or not at all virulent on our host. Um, I'm hoping that we can start collecting more natural populations. We're looking at sort of individual strains, so we can get more natural populations and see some of that variation and start making sense of the 50% successful wasp, because we don't have that one yet. So that's something that I would really like to get into. But yeah, the flies have all the goods to do a successful immune response. It really comes down to if the wasp can outdo them or not. Yeah, yeah.
Without the emit, so kind of a cycle of wasp populations get bigger, fly populations get smaller.
Oh, that's a cool question. So, short answer is I don't know. Slightly longer answer is kind of. So, we at least know very early in the season. So, when flies start waking up from winter time, there's a time when there are no wasps and there's lots of flies. Once the wasps start waking up, you start now seeing that total number of insects is more split between flies and wasps. I don't know sort of like say throughout the season how cyclical it is, or if it now reaches an equilibrium where wasps are winning X% of the time. But there's early on, the flies have a definite advantage when there's no wasps out of hibernation yet. And then as soon as the wasps come out, they assert themselves. Yeah. Oh, that's a good question. So we have not looked at this cellular response temporally throughout the day. There is a lot of work looking at like antimicrobial responses, which do have peak times, which seem to be tied into the circadian clock. Don't know if that's the case here or not. And so we've never really looked at the wasps for when they're most active. That would be that would be an interesting thing to do, to sort of set up like a big population and see if there is something timed happening throughout the day. We've never noticed, but I don't think we've done the experiment that would let us notice. All right, any other questions? Yeah.
Said sort of this plastic response, you sort of have a generalist multiple generation.
Yeah.
So what's the cue there? Is it developing in that thing, or is it, because that seems pretty spectacular?
Yeah, no, it's a fantastic question that I literally don't have an answer to. That was a student that started right before I left Illinois to come here, and he could not move with me, so it was kind of a long distance, and we didn't get as much into details as we wanted to. I mean, he did a really good project and did some other stuff and has a real job now, but yeah, there's a lot about that I would love to go back and look at more closely. Yeah.
Well, question.
I'm curious, like, Jack, because you were mentioning that software is a great comp.
Yeah.
Human thing that like we humans care about.
Yeah.
If you were to scale this up, like in the 10,000 foot view of this work.
Yeah. Yeah, so that's a great question. I think, to my mind, there are two very distinct parts of it. So we'll call the first natural product discovery, right? And I like to think of these venoms as essentially a mix of weirdly evolved proteins that do cool things. And so I think we can use this as a way of start looking at applied functions, right? So we have fly models of different types of cancers, different neurodegenerative diseases, and we're finding venom proteins that are able to inhibit signaling pathways that contribute to these diseases, right? So we have like fly cancers and fly leukemias, and we can cure them by infecting them with a wasp. I mean, the wasp eats them, so it's not a great cure, but on some level, we're manipulating pathways that are important in a disease context. And so I think we can use this to start maybe understanding principles about how we can tackle manipulating signaling pathways that are not very accessible so far. I also really like these vesicles, and I should have spent 3 hours talking about the vesicles, but one of the things that's cool about it is not only are the proteins appear to be packaged in different vesicle types, they get sent to different tissues in the host. So we have the SERCA that I was telling you about that changes calcium signaling. I looked at calcium signaling in a whole bunch of other tissues, and it doesn't get changed anywhere but immune cells. And we have this epithelial tumor model where the venom blocks the growth of the tumor, but it doesn't block the growth or the division of any other cell type that we can see, only the tumor and the precursor cells that are dividing at the same time. So we have these very, very specific, what appear to be very specific targeting mechanisms that I think we can try to figure out the principles about how to target protein cargoes, and that could have some applied function. The other side of it is immunity. So I'm not an immunologist. I've never actually even taken a class in immunology, but I think it's so cool when you can look at things like activation of cells. I didn't talk about it at all today, but we have flies that mount an autoimmune response. So we're understanding how innate immune cells can do self-tolerance. So kind of all these weird questions and understanding innate immunity are things I think we can study with this, because we're looking at a naturally evolving system and it responds in ways that we can predict, kind of thing. Yeah. All right, excellent. Thank you.
We'll also be heading over to Bombs for happy hour for anyone who wants to join us.
Yes, I am very